This lecture will be on blood and its components. So blood evidence can be used to do a few different things. It can be used to indicate the type of weapon that's used because we can look at the spatter patterns and see if there was a high velocity or low velocity wound inflicted, such as a gunshot versus, something with a baseball bat. We can also use it to recreate a crime scene because we can use the spatter to actually locate where the victim was and where the assailant was, if they were standing, if they were on the ground, if they were at an upward angle, or if they were lying on the ground and shot up. And then we can narrow down a suspect to a group because blood is class evidence. We can figure out, what percentage of the population they're part of, depending on if they have A, B, or O type blood. So blood composition is made up of red blood cells, which are called erythrocytes, and that's what these are up here. They kind of look like little tubes with centers, and they carry respiratory gases, so oxygen and carbon dioxide. Remember that oxygen will be in the lungs. The red blood cells will have that oxygen, move it to all the cells of the body. They'll pick up the carbon dioxide and bring it back to the lungs where we exhale it. And hemoglobin is what gives blood its red color, and that's the substance that actually binds to the oxygen. White blood cells are also called leukocytes, and these are all different types of white blood cells. And their job is to fight diseases and foreign invaders. And they will, some of them are actually antibodies, and these are proteins that are secreted by some white blood cells, which assist in the immune response. Platelets or thrombocytes, these little guys here, are actually fragments of blood cells, and they aid in clotting and repair of blood vessels. So when you get a scab, what stops the blood flow, when you cut yourself, the platelets will actually go to that site and start secreting proteins, which will cause red blood cells to be trapped in a web, and that will form your scab eventually. And the liquid component of blood is plasma, and its composition is similar to salt water, and its function is to carry dissolved proteins such as antibodies and hormones, as well as nutrients like glucose and salts. If we take a look at this centrifuge tube here, this almost looks like a tube they would take your blood from, and this would all be read at one time. They'll put it in an instrument which is called a centrifuge, and a centrifuge spins really, really fast. And when it does that, the components in this tube will actually separate by density. So the red blood cells are the densest, and they'll settle to the bottom. And then you have your platelets and white blood cells. And then the plasma, the liquid part, will settle on top. So then the lab technicians can, take out the components that they need to look at, to figure out, what kind of illness you have, or if you're, missing some sort of nutrient in your blood fluids. And a little history on red blood cells and white blood cells. In 1982, Sir Alec Jeffreys actually used white blood cells to produce the first DNA profile. Way before that, in 1900, Carl Landsteiner found that blood from one person did not freely mix with blood from another person, and he described the A and B proteins in 1901. So these proteins are actually, remember that we have A blood and B blood, but these are the only proteins that you'll find. You might have both proteins and have AB blood, which we'll take a look at later. And there's a picture of some rhesus monkeys down here because later on, which we'll discuss, there was another protein that was found, which was called the RH protein, and it was found in the rhesus monkeys. So that's what's actually going to give you your positive or negative blood type.